Good afternoon, everyone, and welcome to session 4 of the symposium, in which we will be focusing on innovations in pediatric cardiovascular medicine. This is an important area of focus for the Maternal and Child Health Research Institute, which has funded many projects in this area, including a directed effort to support research in single ventricle disease. My name is Melissa Mavers, and I am an instructor in the Division of Pediatric Hematology, Oncology, Stem Cell Transplantation, and Regenerative Medicine. And I will be introducing our speakers and moderating questions. Our first speaker is Dr. Casey Gifford, Assistant Professor of Pediatrics in the Division of Cardiology and, by courtesy, of Genetics, who will be presenting her work on the genetics of congenital heart disease. Dr. Gifford.
Thank you for that introduction. I wanted to start by thanking MCHRI for giving me the opportunity to tell you about our work today. So heart development is governed by a series of complex morphogenetic events that occur largely during the first few weeks following fertilization. And in my lab, our focus is on understanding the molecular mechanisms that dictate these events. And one reason for focusing on this area is the incidence of congenital heart disease. So congenital heart disease is found in 1% of the population, making it the most common congenital abnormality. And congenital heart disease is actually an umbrella term for more than 35 subtypes of structural defects that an individual could have when they're born. And interestingly, if you're diagnosed with congenital heart disease, you're also at an increased risk for other defects and diseases such as neurodevelopmental delay and pediatric cancers. And lastly, congenital heart disease has a high recurrence rate in first-degree relatives, which actually tells us that it's likely genetic in nature. And while it's genetic in nature, or we believe it's genetic in nature, and this has been understood for quite a while, genetic diagnoses in this area are quite difficult. And so to this day, given all of the studies, exome and whole genome studies, sequencing studies that have been done on CHD cohorts, more than half of CHD cases cannot be solved. They have no known genetic etiology. And there's a number of reasons why that might be, including a lack of understanding of the genes in heart development, so prioritization of variants is difficult. Our ability to appreciate non-coding variants is really difficult, which could complicate diagnoses. And then finally, there's this idea of oligogenic mechanisms or multigenic mechanisms of disease that may explain some fraction of unknown cases. And this is what I'm going to focus on for the remainder of my talk. And so what is an oligogenic disease? What do I mean when I say that? And so here I'm showing you a very traditional effect size by frequency plot, where the frequency of a variant is on the x-axis and the effect size is on the y-axis. And on the left side of the plot, you have variants that are extremely rare or perhaps private, and they tend to explain monogenic diseases. So these are diseases where a single variant has a large enough effect size to explain a phenotype or disease. And then on the right side of the plot, you have polygenic disease. So these are diseases or phenotypes whereby many variants have a tiny effect size, but collectively contribute to a phenotype. And height is a really good example of a polygenic phenotype. And then in the middle is another type of complex disease, which I'll refer to as oligogenic, where you might have a small combination of variants where individually their effect size is small and insufficient for a phenotype, but in combination, they would be sufficient to lead to a phenotype. And historically, it's been very difficult to prove the variant involvement in these types of mechanisms because we lacked a lot of the experimental and computational tools. And we've thought, as I mentioned previously, that congenital heart disease might actually lie in this middle tier, and we just haven't had the tools to pursue this previously. And there's a lot of evidence to support this idea. And I just wanted to point out one reason why we thought that oligogenic inheritance might contribute, or oligogenic mechanisms might contribute to congenital heart disease, is pleiotropy when it comes to cardiovascular phenotypes. And so historically, when we would prioritize variants in patients with congenital heart disease, there are a number of genes such as NKX25 or Notch1 that are associated with variants in these genes are associated with cardiovascular phenotypes. But the issue is that you would find different phenotypes associated with variants in these genes. And so that tells us that this gene may contribute to a phenotype, but it is likely not sufficient for the ultimate phenotype that we see. And this is just one example of why we believed there was more information contained in one's genome that might explain defects and disease. And so when I started my postdoc, we wondered if we had new tools that would allow us to move beyond genetic associations and really provide experimental support for oligogenic diseases, and tools that would allow us to study the specific variants of interest in better model systems. And in order to ask this question, we decided to focus on this one familial case that included a family with heart disease with variable symptoms that came to our attention when this individual in the pedigree here presented in congestive heart failure at 1 week of age, requiring mechanical ventilation and inotropic support. And this individual was ultimately diagnosed with a defect called left ventricular noncompaction. And so here on the left side, I'm showing you an echo image of an unaffected heart, where you have the left ventricle here in this image. And I'd just like you to focus on the free wall and the apex of this heart is relatively smooth based on this echo image. However, when you look at the echo image of the affected individual, you'll see where these yellow arrows are these protrusions into the left ventricle, and these are called trabeculi. And it is these protrusions into the left ventricle that led to the diagnosis of something called left ventricular noncompaction. So what is left ventricular noncompaction? It's a defect that we think arises when there's a delay in myocardial development because trabeculi protruding into the left ventricle are present in embryonic development, and eventually during maturation, they compact into a smooth left ventricular wall. It can be isolated or complex, meaning you can find it in combination with other defects. It has an extremely variable clinical presentation. You might actually be walking around with a hypertrabeculated left ventricle now and you have no symptoms and you don't know it, or you could present in heart failure at a very young age. And genetic associations with this defect have been very difficult. So there have been variants in a number of genes associated with this defect, but they're usually not completely penetrant. And so it's been difficult to predict who would have this disease ultimately. And down here, I'm showing you cross sections of human hearts where this is, and that are age matched. And here you can see that in a healthy heart, you would expect a relatively compact, smooth left ventricle, whereas here you can see that there's a mesh-like network that represents these protrusions that stick out into the left ventricle and really prevent the heart from functioning properly. And when we took a further family history, we found that, well, this was the original individual that presented. This individual had two siblings that also exhibited symptomatic LVNC. And then a father that upon imaging had protrusions in their left ventricle, but was asymptomatic. And so this was where we thought that this would be a good test case for understanding oligogenic inheritance, where our hypothesis is that there are variants encoded in the genomes of these two parents that are contributing to the ultimate phenotype that they see in all three children. And so the first step in asking this question was to perform exome sequencing. And so we asked two hypotheses of the exome sequencing data, which I've highlighted here. The first is to ask and look for variants that are inherited from the father by all three children. And this is a fairly traditional exome sequencing prioritization scheme, including a filtering step where we're looking for variants that are damaging, single nucleotide variants that are damaging to the encoded proteins. And we identified two single nucleotide missense variants of interest, one in the transcription factor MKL2, which is a myocardin-like transcription factor, which has never previously been associated with congenital heart disease or heart disease in general previously. However, the mouse knockout does lead to a hypertrabeculated left ventricle, which was very exciting to us. And then we did identify a variant in the gene MCHRI, which encodes a protein that's essential for sarcomeric formation and function. And mutations in this gene have widely been associated with cardiomyopathies and congenital heart defects, and it's no surprise that this variants in this gene contribute to heart development. But because this variant was in all three children and in the father who was asymptomatic, we did not believe that it was necessarily sufficient for disease, which was why we went on to continue to look for additional variants that might contribute to the ultimate phenotype. And the second hypothesis we asked was, were there variants that were encoded in the mother's genome that are inherited by all 3 children? And this is a situation where we're looking for modifiers of the disease, because the mother is fine. Her echoes, her imaging, her MRIs show no symptom or no defects in her left ventricle. She has no symptoms. So we looked at this data a little differently in that we removed this filtering step related to damaging variants, because we didn't believe that these predictions were necessary for a modifier, because it's not sufficient on its own to lead to a phenotype. And this analysis was also interesting to us because we identified a variant in the transcription factor NKX25, which I introduced to you previously. And this is a transcription factor that's essential in the very early stages of cardiogenesis that has previously been associated with CHD and cardiomyopathies. And this variant is out there in the population in around 1 in 1,000 individuals. And so this is what we think of when we think of a modifier, right? It's a population that's circulating out in the population, and on its own, not sufficient. But in the unlucky event that it's combined with other deleterious, slightly deleterious, small effect size variants that collectively they're leading to a phenotype. And so I'm going to summarize many years of my life in just a couple of slides and say that, and first highlight some of the mouse work that we did. So to really understand whether these variants were contributing to left ventricular development and perhaps the LVNC phenotype, the first thing that we did was generate mice that harbor the orthologous variants using Cas9. And CRISPR-Cas9 tools are really what I was speaking to when I said, do we now have the tools to move forward and provide experimental evidence for specific variants being involved in multigenic diseases? And here I'm just going to share with you the answer to the ultimate question, which is, if we have a mouse that is a triple compound heterozygote mouse, similar to the patient's, would it exhibit abnormalities in the left ventricular free wall? And the answer is, of course, yes, or I wouldn't be telling you about this story. So here I'm going to show you cross sections, 4-chambered views of P3 mouse hearts, so 3 days after birth. And in green, we're highlighting just the structure of the heart. And then red represents antibody staining of endomucin, a gene that is expressed by the endocardium, which lines the inner chamber of the left and right ventricle. And so you'll notice that in wild type mice on the top left, especially down near the apex, we have a very few protrusions. The left ventricle is fairly clear of protrusions. When we look at an NKX25 heterozygote, however, we start to see that there are more trabeculi or tissue protrusions into the left ventricle. And then when we look at the double mutant, the MKL2 and MYH7 double heterozygote mouse, we see something similar where we see these protrusions into the left ventricle. And then finally, when we look at the triple heterozygote mouse, we see protrusions into the left ventricle, as we were seeing in patients. And we also saw a very thin apex with protrusions all the way down and into the apex. And I just wanted to zoom in to this image and show you this is the apex from that mouse. And then to say, I'm highlighting this image because often in textbook examples of LVNC, you see characteristic depictions of LVNC occurring in the apex and really seeing these protrusions into the apex, but then also a very thin apical wall. And that's indeed what we're seeing in our mice that carry each of these 3 variants. And we can, of course, quantify this across many mice from many litters, which is what we did. And so here on the left graph, I'm showing you apical thickness. And so you can see that as we move to the right, we're increasing the number of variants in the mice, and you can see that the apical thickness goes down. So showing you that we're seeing that in many mice, that there's a very thin apical myocardial wall. And then we can also quantify the level of trabeculation, which we did using fractal geometry. And so you can see that the complexity of the left ventricle based on the endocardial staining is indeed going up as we move to the right in this graph. Again, showing that as we add variants in these mice, as their genotype becomes more complex, we're actually able to recapitulate the disease and able to see that each individual variant is indeed contributing to the thickness of the myocardial wall and to the level of protrusions into the left ventricle, similar to what we would have expected from the patients or seen in the patients. So this provides some histological support that these variants are contributing to LVNC. We additionally wanted to provide molecular support. So we performed RNA sequencing of these hearts. And indeed, we found that in mice that are triple heterozygotes, we found a number of genes that are expressed more highly in these animals that are associated with cardiomyocytes in more immature stage of cardiomyocyte development, suggesting that these hearts are indeed more immature, as has been predicted for the disease cause, that there's a delay in myocardial development. And so this RNA sequencing would suggest that. And then excitingly, we also saw the downregulation of a number of genes that are important in endothelial and endocardial development. And this was exciting to us because there's been work done by many, including Christy Redhorse here at Stanford, to show that disruption of endothelial cell and endocardial coronary vasculature development can contribute to LVNC, and it's the deletion of these genes that leads to this phenotype in mice. And finally, we can also take patient-derived iPS cells and use these cells in a differentiation system to recapitulate cardiovascular development. And this is the obligatory video of beating cardiomyocytes taken by Emily, a member of my lab, to show you that we can recapitulate development to some extent in a dish using patient-derived iPS cells. And in taking some iPS cells from these individuals and performing RNA sequencing on cardiomyocytes derived from their stem cells, we saw something similar in the dish, which was that genes associated with earlier stages of development were expressed at higher levels in the most affected individuals, suggesting again in vitro in a cell autonomous fashion that these cardiomyocytes are also more immature than you would expect. We also, through a series of expression analyses across a time series, could show that indeed this is a delay in differentiation. So BMP10 is a marker of cardiomyocyte differentiation, and you can see in the individual that was affected by LVNC, the peak of BMP10 expression is occurring later in differentiation compared to the unaffected individuals, again suggesting that not only is this defect a delay in differentiation, but that we can model it in both mice as well as human iPS-derived systems. So in summary, we've recapitulated a human phenotype using single nucleotide variants. So these are, again, not straightforward knockouts. These are orthologous variants found in humans. We believe we identified a novel congenital heart disease-associated gene in MKL2 and a genetic modifier in NKX25. And we really believe this is the first experimental support for oligogenic inheritance of CHD. And so now, just quickly, in my lab at Stanford, my goal is to make precision medicine a reality for individuals and families impacted by CHD. I wanted to introduce you to Tatiana and Anna. These are the two women that I was using in my study, and I'm very grateful for their involvement. And my goal is to make sure that we're promoting precision medicine for them so that when they want to have children, we can be a little more informative when it comes to genetics. And so we're broadly interested in defining modifiers at scale. And so we're performing and still pursuing case studies of individual families. We're also pursuing genetic interaction studies to better understand and perhaps predict which genes interact so that we'll be able to look at oligogenic scenarios and predict which combinations of genes are important. And then we're ultimately also interested in population genetics, because the sequencing of everyone's genome is really going to help us better understand how all these variants contribute to phenotypes. And I wanted to put in a plug that towards this effort, we are expanding our biobanking and whole genome sequencing efforts in the Children's Heart Center. And so that's something that we're really excited about that will facilitate our research. So I'll just stop by thanking my former advisor, Deepak Srivastava, all the members of his lab that helped me do this work and gave me the freedom to do such an exciting project that could serve as a foundation for my new lab here at Stanford. And I'm very thankful for all of your attention, and I'm happy to take questions.
Thank you, Dr. Gifford, for such an excellent talk. Really an ingenious approach to using the children along with the mother's and the father's genetics to elucidate the combinatorial effect. And as you alluded to, a huge amount of work to recreate that in the mouse. So we have a couple questions in the chat. I think we might have time for one of them, which was, is the practice of filtering against common alleles, especially non-coding alleles, hampering our detection of determinants of oligogenic heart and other disorders?
I think so. Yeah, I absolutely think so. I think it's very hard to have evidence or to understand the context of the importance of common variants. Because they're common, we inherently know they're probably not sufficient. So then the question is, what else is contributing to the phenotype? And that's often a very hard question. You have a common variant, but then you're also going to have hundreds or thousands of additional variants in that genome. And so how you next prioritize the other variants involved is a very complicated question and something I'm very interested in understanding.
Right. Yes. I think we maybe do have time real quick for the second question. Do you think sex differences contribute to the presentation of the family offspring, i.e. male had worse outcome?
It's very possible. I don't have a good hypothesis because what I didn't mention is the individual that passed away, that happened many years before the little girl we studied presented. And so this DNA had been in a frozen OCT block for many years, and the quality of the DNA wasn't great. And so it's hard for me to identify additional genetic variants. All I could do was confirm that he carried the ones I was interested in. But yeah, I think a sex difference might explain that. It's very possible.